Hello students, this is Lázaro Pino, your chemistry professor. Today we are going to have Introduction to Carbohydrates and its classification. We can say that 50% of the carbon in organic compounds is found in carbohydrates. Through photosynthesis, plants store energy in form of glucose, a simple sugar. They take carbon dioxide from the environment and water from the roots, and using the energy of the sun, they produce glucose, and at the same time, they release pure oxygen to the atmosphere. The opposite to photosynthesis is the cellular respiration. Animals used to take the glucose during a combustion reaction, they produce carbon dioxide, water, and at the same time they form ATP that is necessary for all the activities in our body. We can classify the monosaccharides into aldehyde functional group, the aldoses, and the ones that contain the ketone functional group, that are the ketoses. Here you have, for example, that glucose and ribose contain the aldehyde group, then they are aldoses, while fructose contains the ketone group, then is a ketose. We also can classify the carbohydrates into aldoses and ketoses, and according with the number of atoms of carbon. In this case, you have 1, 2, 3, 4, 5, then it is a pentose. But because of the aldehyde group, it's also an aldose. In this case, you have 4 atoms of carbon, it's a tetrose. And because of the aldehyde group, it's an aldose. In this case, you have 3 atoms of carbon, it's a triose, sorry. But also is a ketose because of a ketone group. Here is another form of classification of simple sugars according with the position of the OH group in carbon 2. In this case, that is a chiral carbon in glyceraldehyde. We can divide the glyceraldehyde into D or Al. D in this case, because is to the right, the OH group, and in this case, to the left, that's why acyl glyceraldehyde. Usually, what we do in order to study the carbohydrates is to make a plane or to get a projection of the molecule in the space. And this way, we can use this projection, that is a Fisher projection, to study better the structure of carbohydrates. Here we have D-glucose and L-glucose. The further chiral carbon to the aldehyde group is used to classify glucose into D with the OH to the right, or L-glucose with the OH to the left. Also, we have the fructose, that is one of the products that makes 40% of honey. Two important sugars are ribose and deoxyribose. They are found in RNA and TNA. Here we have an example of a monosaccharide derivatives. D-glucosamine is an example where the OH in this position is changed by an amine group. By hydrogenation, we can reduce the aldehyde group to an alcohol group. And in this case, we can convert glucose into sorbitol. This is another example of derivatives. Benedict's reagent that we have seen in the lab is a reagent made of copper 2. that because of the oxidation reaction that is happening in the aldehyde group of glucose or other monosaccharide is converted into a carboxylic acid group, and the copper goes from 2 to copper 1. In this case, we say when the sugar is positive to benedict, that is reducing sugar. Here is another representation of sugar and the real one. We join, in this case, the carbon 1 with carbon 5, and we make a hemiacetal. Is a cycle that is represented, as you can see here. This is the actual way glucose is in solutions. This is the Haworth projection that is real, is a real one, and not the Fisher projection that is used to do studies and is easier to be understood. When we are in the cycle form, we can have what we call a numeric hemiacetal. The carbon 1 in this case for glucose makes the difference between two glucose. Alpha if the OH in carbon 1 is down, and beta if the OH in carbon 1 is up. The union of two monosaccharides produce a disaccharide. Here we have maltose, that is when we join together two glucose units by a glycosidic bond. Lactose is made by the union of galactose and glucose by a glycosidic bond, and the type of this bond is beta 1,4. Sucrose is also the union of two monosaccharides, glucose and fructose, and they are joined by a glycosidic bond also. Sucrose is the table sugar that we use at home. Generally, we use sucrose as the unit, and we give a value of 100 to is relative sweetness power. Then you can see here different sweeteners, but they are natural. And here we have certain artificial sweeteners. As you can see, we don't find any unit of sugar in it. They are organic compounds that may produce a lot of sweeteners, of sweetness in the food you eat, but as they don't produce glucose, you know what is going on. That because of homeostasis of blood in sugar, when the level of blood glucose is low, the alpha cells release glucagon, and then through glycogenolysis in the liver, the sugar that is stored in form of glycogen released glucose to blood, and you get the normal blood glucose. But if it is very high, the level of glucose, then the beta cells in pancreas produce insulin that binds glucose and take them into the cells to produce the cellular respiration and produce ATP. This way is that we get the homeostasis of blood glucose. If we use a sweetener, we are not going to get any type of sugar, and then we are going to need to eat more and more food in order to obtain the level of glucose that we need. And at the end, we have for example, in aspartame, side effects, as you can read here, that are not very healthy. Then, don't eat any kind of sweeteners. They don't produce glucose, and they have, by the way, side effects. Then, the the cellulose is a polysaccharide that is made by many units of glucose. They are joined by beta-1,4 glycosidic bonds. But between the chains, there are another type of bond that are the hydrogen bonds. That's why good contains 50% of cellulose, and that's why it's so strong. Starch, like amylose, is made of linear chains composed by glucose units. The bonds between the glucose units are alpha-1,4, and it is not linear like cellulose is coiled. The other starch is amylopectin that contains branches made by glycosidic bonds of the type alpha-1,6 to the normal ones that are the alpha-1,4 glycosidic bonds in the linear chain. Then amylopectin is branched. Similar is glycogen, that is the way animals store glucose. But as you can see, there is a difference between glycogen and amylopectin, and glycogen is more branched than amylopectin. Another type of polysaccharide is chitin, that is in the exoskeleton of certain animals. They are very important because they are used for several medical devices, drug delivery, and catalysis. As you can see, chitin is made of units of glycosamine that we have seen before, where one chain in a side is made of a nitrogen group. Well, in general, we can classify carbohydrates into sugars that are monosaccharides and disaccharides, and polysaccharides that are made according with the function of them, they can be divided into storage and structural. Storage is glycogen and starches, while structural are cellulose and chitin. Well, that's all for today. See you in the next